see. There you go. Just to give a little bit of background, one thing we're always struggling with when you're trying to image anything, and this applies to microscopes, but it also applies to any kind of imaging device, including a camera, is what we call the eternal triangle of compromise, where we are constantly struggling to balance the sensitivity or the resolution or the speed at which we can acquire images. So if anyone's ever used a manual camera before, they may be familiar with the scenario where they want to capture a scene with their camera, and let's say it's dark. Then one option you may have is to increase the exposure time on your camera to let in more light. Now you could do that, but that comes with trade-offs. For example, if you increase the exposure time, that means your imaging is slower. So if you're trying to capture a moving object, it's going to be blurry. And it doesn't matter how sharp your camera is. If you're doing a long exposure and it's blurry, you could capture 1 million pixels of a blurry object. It's not going to let you see any more detail in that object. So in reality, speed and resolution are actually intimately coupled when it comes to moving objects. And the reason you need to do these long exposures is because you don't have enough sensitivity. So another option is you can increase the sensitivity of your camera. And if you've ever used a manual camera, you'll know there's something called the ISO, which is, in other words, gain of the detector. So you can increase the gain, which means that it'll be much more sensitive to every single photon that comes in. But that also comes with a trade-off, which is more noise. So as you increase the gain of the detector, it starts to become a noisier image. And that's what you usually see. If you take a picture with your iPhone, for example, in a dark room, you'll notice it looks really grainy and blurry. It doesn't look like a nice quality image with as much detail as you would if you had full light, like an outdoor photo in the middle of the day. So that scenario of constantly trying to balance the amount of light you have and the amount of sensitivity and resolution and speed also applies when you're sitting at the microscope trying to image really small stuff. And it sucks because we'd love to have maximum sensitivity, resolution, and speed so we can get all of that juicy detail out of our experimental samples. That's just not what reality allows. So I'm going to show some examples throughout this talk of what that really looks like at the microscope. So here's an example right now. Hopefully you can see this well enough on your computer screens. On the left and on the right, we're looking at the exact same sample. On the left, what we're looking at is that sample being imaged with 8 nanometer pixels. And on the right, they're being imaged with 2 nanometer pixels. Now, one of the first things you should be able to tell is that when we're imaging with 8 nanometer pixels, the screen is refreshing much faster. We can image much faster. This is using something called a scanning electron microscope, which is one type of a point scanning imaging system. The way they work is you scan your electron beam over the sample in a line as a raster, 1 pixel at a time, kind of like a typewriter, moving down, line by line. And as you can see on the left, we can do this really fast. On the right, we're imaging with 2 nanometer pixels, and you can see it's 16 times slower to raster through. That's because we're imaging 16 times as many pixels. The pixel size, pixel is XY. And so when you go from 8 to 2, that's 4, but you have to do it 4 times 4, so it's 16 times slower. But what you can also see in the image on the right is that it's sharper. We can see more detail in this image on the right than the one on the left. And it's also higher signal to noise. The contrast is better. And what we're actually looking at here is a presynaptic photon. This is a fundamental unit of the brain where all those neurotransmitters are housed, ready to be released across the synapse. So those little circles that you see, I don't know if you can see my mouse pointer, these little circles are presynaptic vesicles. And one thing we want to do when we're imaging brain tissue is to understand the distribution and size of these presynaptic vesicles. And as you can see in the image on the left, when we're imaging much faster, you can hardly see whether something is a vesicle or not, or it's just some smudgy dirt. Whereas on the right, we get a better chance at actually being able to see that. And just to highlight the practical importance of being able to image faster, like why would you ever image with the resolution on the left versus the resolution on the right? Well, the reason is if you want to image a lot of tissue, let's say you wanted to image an entire brain from a mouse, which is a pretty small animal. To image it at the resolution on the right with a higher resolution, 2 nanometer pixels, it would take about 160 years. But if we can do it 16 times faster, then we could, like on the left, then we could image for 10 years. That's still a long time, but 10 years is something we might actually attempt to do. It's very hard to convince people to start a project that'll take 160 years. So these are real world trade-offs and decisions that we make all the time. So there's a lot of people imaging with the resolution on the left simply because they want their experiment to be finished before they die. Another example of trade-offs is on the left, we're imaging with high resolution this time, and on the right, we're imaging with low resolution. And what we're doing is we're imaging with something called a serial block phase electron microscope. The way this microscope works is it images the tissue, then it cuts off a very thin slice, and then images the new surface, then cuts, then images, and cuts an image, and cuts an image. This allows us to do more high throughput electron microscopy. It's a wonderful machine, cost us $1.5 million to purchase. But what they don't tell you when you buy the machine is that if you want to image with really high resolution, like on the left, unfortunately, those electrons, you need so many more electrons to image at high resolution because you're imaging so many more pixels. Those electrons start to melt the sample. And when the sample melts, whenever you try to cut it, it doesn't cut cleanly. It starts to smudge. And that's what you see happening on the left. The sample is getting smudged during the cutting process, and it's getting destroyed. Whereas in the image on the right, because we're using 16 times fewer electrons, the sample is cutting just fine, and we can slice through and image through that whole piece of tissue. However, as you can see in the image on the right, we once again have lower resolution, lower contrast, lower signal to noise. So we're able to image, but we can not get as much detail as we would like as if we could image with the resolution on the left. So I was banging my head against a wall dealing with these issues when I happened across this article, October 2017, in a photography website that talked about how you can use AI to enhance low resolution photos. And on the image on the left, you can see at the low resolution. On the right, you can see the output of the AI. And I was just blown away. I've been thinking about AI for imaging, but I was thinking about it more as a tool for detecting and classifying objects. I hadn't thought about it in terms of image restoration. So that led to the obvious questions. Can we generate a deep learning super resolution model that restores undersampled images acquired on a point scanning microscope? Because if we could image, as you saw, at lower resolution, we could image faster. We could image on our special microscope. And then if we can use deep learning to restore those images to full resolution, then we can get the best of all worlds. We can image fast, we can image without destroying the sample, but we still get all of that juicy detail that we love so much. So this is the strategy we came up with. We actually use semi-synthetic training data to train what's called a UNet, to be able to take low-res data and convert it to high-res data. So what is semi-synthetic training data? What we did was we got a hold of about 160 gigabytes of gold standard electron microscopy data acquired by Kristen Harris's lab, a collaborator at UT Austin. And then we developed something called a crappifier, very technical device and term, very formal. And what this crappifier does is it transforms the high resolution data into low resolution data that looks a lot like the low resolution data we acquired on our microscope. And then we use these image pairs to train our UNet to go from this low, crappy version back up to the high version. And then we use that UNet that we had trained and applied it to real world data where we actually acquired it at low res and at high res, and then compared the output of our model to the actual high res data. Now, why didn't we just acquire lots and lots of low and high res data and train a model like that? Why did we use a crappifier instead? Well, what some computer scientists may not appreciate is that acquiring these images is extremely time consuming and difficult. Even just acquiring the high res data is time consuming and difficult, but acquiring image pairs at high and low res is extremely difficult. First of all, you have to take twice as many images. So you're talking about twice as long of an imaging time. Second of all, those samples, when you're imaging them, you're bombarding them with electrons or photons. You have vibrations in the building. You have vibrations in the instrumentation itself. So by the time you finish acquiring a high resolution or a low resolution image of your sample, and then you move to image it again at the low resolution or high resolution, whichever one you're doing first, the sample has moved and changed. Not a lot, but a little bit, which means now you've got image pairs, but they're not perfectly registered. They may be distorted. They may have moved in and out of focus. And that's a random process that you have to somehow correct for. And to be able to do all of and get many, many image pairs is just incredibly time consuming. So this table shows you if we had to do real world data pairs with, let's just say 80 gigabytes of EM data, it would have been 480 imaging hours. And in my core at Salk, where our rates are extremely cheap, this is not how much it really costs, this is just how much you would have to pay, it would still be a $17,000 endeavor. However, if you're like us and like many other people in the world, if you can access data that was already acquired at high resolution and then just crapify it, all you need is 2 hours of any decent computer to crapify 80 gigabytes of data. And that would only cost $16. And then, of course, if you don't already have high-res data, you can just image the high-res data alone, crapify it, and you'll still be better off. You know, it'll cost half as much. So that is why we developed the crappifier. Now, it's not necessarily trivial to make a crappifier that actually works. And what I mean by works is that you train a model on crapified data, but it works on real-world data. Because the concern is that if you use a bunch of synthetic data for training, your AI is going to learn how to hack your artificial manipulations, but it still won't succeed when it deals with real world data. This is always a concern. So for us to develop our crappifier, we actually compared a bunch of different crappifiers. And so the first thing we started with was, what if we just downsample our data? So here's the ground truth data. This is high resolution image, ground truth. And if we just downsample the pixels without adding any noise or any other manipulation, then this is what the data looks like. And if we train a network on that, you can see what the output looks like. Doesn't look very good. Still looks better than the low-res data, but it doesn't look as good as the ground truth. So then we said, okay, well, we know that there's a lot of Poisson noise in microscopes. What if we add Poisson noise? So we add Poisson noise, and that's what this looks like. It looks like the low-res, but it's got some added noise to it. And then we train a model. It doesn't actually look that much better than not adding any noise at all. All right, what about Gaussian noise? We know there's a lot of Gaussian noise in images. Here you can see it's quite degraded compared to just no noise. And then we looked at the output and it actually did worse. So then we said, well, what about additive Gaussian noise? Now, what additive Gaussian noise does is it adds more noise wherever there's more signal. So if there's no signal, it's only going to add a tiny bit of noise. But if there's a lot of signal, it's going to add a bunch more noise. And it's going to add it in a distribution around the pixel with the signal. So we did like a sigma of 3 neighboring pixels or 5 neighboring pixels. I can't remember which one. We used one for one and one for another. But anyway, when we did the additive Gaussian noise, again, you can see this is what it looks like. The noise is not as distributed as a Gaussian. You can see that our output actually looked pretty good. And then we actually trained the model with the real world training data. And you can see that it did about the same as the additive Gaussian noise. But then here's the real trick. To use real world training data, we couldn't acquire 80 gigabytes of data. We could only acquire like, 10 gigabytes of data, if that. So then we asked, what if we did additive Gaussian noise, but we actually used all of that high-res data we had before, the 80 gigabytes of data. Sorry, we did 1 gigabyte of training data for this. And then you can see that the results were significantly better than the real-world training data. And the additive Gaussian data was just 1 gig. But here's what's interesting, and I think this is important for all the students out there, people thinking about AI. If you look at the metrics, these are standard metrics in computer vision for evaluating the quality of performance of your algorithm. PSNR and SSIM, those are pixel-based error metrics. What that means is that they measure the pixel value in image pairs, and they compare pixel by pixel to see how well it did. And the more off they are in terms of gray value or whatever, the more you're penalized. And in both of these metrics, the higher, the better. And what you could see is that the difference between additive Gaussian and real world, either with 80 times as much data or not, they were not significantly different from one another in PSNR. In SSIM, they were a little bit different. All right? But if you look at the image, if you visually inspect it, you can see that they're quite different. So it just highlights the fact that metrics have to be scrutinized themselves, and that there is no single metric necessarily that works for evaluating the output. You cannot just rely on the numbers and the calculations and the statistics to tell you if you're going in the direction you want to go in or not. You have to look at the data yourself. And then what we saw was we added another metric that's not normally used in computer vision, was we measured the resolution of our images. And there we saw that the 80x was much better resolution than the real world or the smaller data set training. So that made sense to us. So here's a closer look at the output. On the left is a low-res image, real-world image that we acquired on our microscope. And on the right is the ground truth, high-res image that we acquired on our microscope. And in the middle is the output of our deep learning model. And a couple things stand out. First of all, we are definitely able now to see these presynaptic vesicles that we could not resolve in the raw data. So that's awesome. Second of all, this doesn't look identical to this. And the reason for that is manifold. But one really interesting thing is that our deep learning model is removing noise that actually exists even in the high-res data, which actually makes it easier to segment. But it also means that we have to be wary of potential artifacts and made up hallucinations. We'll get to that. Long story short, we can take our $1.5 million serial block-based electron microscope now, and we can now use it. We can acquire at 8 nanometer pixels, nice, smooth, and steady, and then run the data through our model and get back all that juicy high resolution data that we wish we could get straight off the scope. Quite happily, it turns out that our model works out of the box, turnkey, without any additional training on other people's data as well, from other microscopes that are completely different from our microscope. So here's data from Jamelia Farms looking at a fly brain with a FibSem, which is a different kind of microscope. And you can see that our model performs quite nicely, quite well on that as well. So that was very cool. And I mentioned that this data is much better signal to noise, which means it's much easier to segment. So we can now pull out all of those details that we want, the presynaptic vesicles, mitochondria, postsynaptic density, ER. Beautifully, this segmentation was done by Sammy Wazanovac, my group. And he said that this was one of the easiest segmentations he ever did because the membranes just really pop out much more easily than they do in normal data. So that's cool. And then finally, we did the hard work of segmenting the data manually with human experts to make sure that we weren't making up nonsense. So again, our metrics look really good. Our deep learning model, PSSR, does much better than bilinear in terms of PSNR and SSIM. But what we need to know is, are the biological structures of interest valid? So we are interested in presynaptic vesicles. So what we did was we randomized images, sent them off to 2 human experts who then counted the presynaptic vesicles in each scenario, high res, low res, after deep learning, and low res before deep learning. And what we saw is that our false negatives, which are in red, went down from 43% to 13%. And we saw that our false positives went up a little bit, from 11 to 17%, which could be interpreted as hallucinations. We're seeing vesicles that weren't really there in the ground truth. However, I need to emphasize that our so-called ground truth data is not perfect. That, first of all, there's noise in it. And here's the most important detail. The error between the 2 humans looking at the ground truth was the same as the error between our deep learning output and the ground truth. So that means that the accuracy is as good as it can get. And that our false positives and negatives may not even be true false positives or negatives. They may just be within the noise of what we can measure in our ground truth data anyway. So that's promising. So I'm going to switch gears and point out that point scanning microscopes aren't just electron microscopes. They're also used for fluorescence approaches, which have unique capabilities compared to electron microscopes. Most importantly, they can be used to image live cells. So here I'm showing you the same thing that I showed you for the electron microscope data in the beginning, which is that if you image at lower resolution, you can image faster, as you see in the upper left. But it's also much noisier. You can't resolve as much detail as if you image at high resolution, like you see on the bottom, high resolution and high SNR. So again, on the bottom, we're using 16 times as many pixels, which means the sample is seeing 16 times as many photons. So we get 16 times as much resolution, much better SNR, but it's much slower. And you may recall I showed you early on how electrons can melt the sample, and that prevents us from cutting. We're not trying to cut our live samples here, but we are trying to image them. And when you image at high resolution, we have another problem with fluorescence of live samples, which is something called phototoxicity. The lasers that we use to image our cells at high resolution are extremely powerful, especially relative to what these cells actually evolved to be exposed to. So in other words, our cells did not evolve to be exposed to high-powered lasers. And when you do expose them to high-powered lasers, they start to suffer something called phototoxicity. As you can see in this video that's playing on a loop right now, in the beginning, the mitochondria are nice, long, and tubular, but towards the end of the video, they start to swell and fragment. And ironically, one of the things we're interested in studying is mitochondrial division. And here what you can see is that the laser itself is cooking the cell and inducing mitochondrial fission. So we're actually perturbing the very process that we want to study when we do high resolution imaging. And we're no longer studying biology. We're studying, as I call it, culinary science. So this is a big problem. So of course, as by now you may have guessed, you can image at lower resolution, like you see in the movie on the right, and then you won't have these phototoxic effects because you're using 16 times fewer photons. It's not as bad. But you don't have as much detail now. You can't really see what's going on. Whereas in a cell on the left, you can see what's going on, but you're bleaching your sample, it's being stressed out by the laser. So again, our triangle of compromise is a problem here. We also have a problem with speed. So on the left, we're imaging at maximum resolution. In this case, we're looking at mitochondria moving along an axon. And on the right, we're imaging the same axon, but in the image on the right, we're imaging at lower resolution, which means that we can capture all of the acceleration and deceleration of mitochondria as they move along the neuron. On the left, we're imaging with higher resolution, but that means that we're only capturing every 4.3 seconds, which means the mitochondria look like they're hopping along. And we can't always tell which mitochondria is which because you lose them and regain them in each frame. Whereas in the movie on the right, you can track them, but then they pass by each other and we can't resolve them because we don't have the resolution. So when mitochondria pass each other, they blend into each other and then they disappear, and you don't know if a mitochondria changed directions or if it just kept going. We have a real problem with trade-offs between speed and resolution to take home here. Reflected in these chimegraphs, where on the left it looks like Morse code, it looks like a dot blob. Whereas on the right, you can see the continuous lines, but they're also very fat, which means we can't resolve individual mitochondria when they pass each other. So we did the same thing. We took a bunch of high-resolution movies of mitochondria and crapified them, and then trained a network to take those high-resolution crappy pairs and restore from crap back to good images. Now, I have to point out, as you saw in all of these movies, these mitochondria are very dynamic. They're constantly moving around. So even if we had infinite money and time, it would be impossible to get image pairs for training if you tried to do real-world image pairs. What I mean is, if you tried to image the same sample at high and low resolution to train a network to go from low to high, you wouldn't be successful, because by the time you finish imaging one of those image pairs, the mitochondria will have moved somewhere else. So when you image the other part of the image pair, you're not imaging the same cell anymore. You're not imaging the same shape or structure anymore. So having this crapifier was invaluable for being able to do this. So as you can see, the results were pretty good. Here on the left is a real-world image acquired in a microscope at low resolution, low SNR, low photon dose. And on the right is the output of our deep learning model. Much cleaner, much sharper. You can see individual mitochondria. This is awesome. But we still had a problem, which is that when you do the time lapse, because of all the noise in this low resolution image, the mitochondria are flickering in and out of existence. And you have to remember that one thing we're interested in is understanding when mitochondria divide and fuse. And when you have this flickering, they're constantly dividing and fusing. Every single frame, you're seeing a break and a joint and a break and a joint. This data is virtually useless for us. So we realized that because this is video microscopy, we could take advantage of that. And what I mean by that is that we have multiple time points that we could use for our training. So instead of training single frame to single frame, we decided to train a multi-frame model where we took time points before and after the frame of interest to help inform and predict on that center frame. With that approach, we were able to get rid of all this flickering. So on the top is a single frame and on the bottom is the multi-frame. And you can see that we have far less flickering, and now we can actually track individual mitochondria as they undergo fission and fusion events. So here you can see a fission event about to occur right here, right there. Whereas in the raw, low-res data, it would be almost impossible to pick that out. So we're very happy with these results. Same thing goes for fast imaging of mitochondria and neurons. On the left, we can image fast at low resolution, but we still can't resolve individual mitochondria as they pass each other. But on the right, after processing through our deep learning model, now you can see the details nice, sharp, and crisp, and we can resolve individual mitochondria as they pass by one another. And we can resolve individual fission events, for example. So, in conclusion, we now have an EM model that is working for EM data from other microscopes. We can get really high quality area scan level images from really noisy, poor confocal images. You don't need real training pairs. You can use semi-synthetic pairs with the proper classifier. This is doable. And we can now do imaging that we couldn't do before. So coming up next, we're going to be building our PSSR 2.0 using some advanced loss functions. I didn't have time to talk about it, but the way our current model was trained is by looking at the mean squared error between the input and output pixels. We're now developing more advanced loss functions that I can talk about a bit. We're trying to improve our classifier, and then we're also moving into 3D models to improve predictions. This work was done by a massive team that I'm really lucky to work with. Lin Jing Feng's the first author. She's our image analysis engineer. She actually is going to be moving on to do a PhD in June. So I now have an open position in my group if you know anyone who would be interested in continuing this work. Fred Monroe is one of the most amazing people I ever met. Really helped us a lot with this project. And Jeremy Howard, of course, very famous, brilliant data scientist, and founder of Fast AI. And Kristen Harris and Lindsay Kirk at UT Austin helped us get the EM data. Sammy Weiser Novak is a genius in my group who does imaging and analysis. It's just a joy to work with. And then we have to thank UCSD's Gulchin Petronas and her student Blenda for their help with the neuronal mitochondria data. And Kara Shirvan's a postdoc in my group who did a lot of the imaging as well. And then Tom Zhang has now moved on to become associate director of another core at Ohio University. But really got to give a big shout out to Lin Jing, first author on the paper, who will be sorely missed when she leaves in June. So with that, I'll take questions, and thank you for your attention. And looking forward to any ideas and suggestions you may have.
Yes, I can ask a question. I was wondering, so I saw on the slide it said you're developing for different tissue samples. So does this model not work on like other tissue samples, say like cardiac or like fibroblasts, etc.?
That's right. Yeah, this is a big problem. If your training data looks completely different from your data that you actually want to apply it to, then it's not going to work. Your training data needs to match. Now, I said that we could use our model on data from other people's microscopes, but the data that they were looking at was still the same type of tissue. So general motifs was similar, But the cool thing is that it doesn't actually need to be identical, which is a misconception a lot of people have, is that if it's not identical, it's not going to work. Certainly not good enough for scientific analyses where precision and accuracy are, at a whole other level. Like this isn't like a deep fake where if the video looks cool and similar enough, everyone's happy. Like it needs to be precise. So if you wanted to do cardiac tissue or other tissues that look quite different from brain tissue, then you would be best served by getting some high-res images of that type of tissue and training a new network. One of my dreams is to figure out a way to automatically classify images and train networks a little bit more automated fashion. Maybe someday train a generalizable model that works for all tissues. But for that, we're going to need quite a bit of coordination and data sharing. And I forgot to emphasize that all of our data, all of our training, all of our code is freely available. And I wish more people did that, because first of all, you're supposed to if you're publishing. Second of all, if people shared their data more, we could work together to train models with other people's data that would be useful to them and to everyone else.
And then my other question was, how are the live cells obtained, like specifically for the neurons? It's kind of interesting.
That's a good question. In our case, the neurons that we were using were taken from embryonic rats. You can dissociate hippocampal neurons from very young rodents and put them in a dish with some nice juicy culture media to keep them happy and alive. And then when you're ready, when they're ready, usually you allow them to mature a little bit more in the dish. You can put that dish on the microscope. We use special dishes that have special glass on the bottom that allows us to do really high resolution imaging with our objectives on our microscope. And to image the mitochondria, there's a couple ways you can label them. One way is to use a genetically encoded marker for mitochondria. By now, we know many different peptides that localize to mitochondria, and you can fuse those peptides to a fluorescent protein, and then you can express that in the neurons. And some subset of neurons will be expressing that marker, and then you can see it in the fluorescent microscope. There are also dyes where you can just add the dye and it'll localize to the mitochondria, and you can see all the mitochondria. And both of those approaches have pluses and minuses. But yeah, that's how we image the neuronal mitochondria. So in vitro, ex vivo system.
No. David asks, could you go more into detail on how your group developed the single frame model into the multi-frame model?
Yeah, sure. You could think of it as a 2.5-dimensional architecture, where we actually kind of hacked it in a bit. So our UNet allows you to have multiple channels. So let's say you had a 3-color image. You could have color, let's say you had an RGB image. You could have red in channel 1, green in channel 2, blue in channel 3. And you can train your network to take an individual RGB images and output a single combined RGB image. So we sort of hacked that structure and simply told it time points -2, -1, 0, +1, +2 were actually 5 different channels. And we use that 5-channel image to make a prediction on a single channel image, which was just T = 0. So actually, I misspoke. It would be like taking an RGB image and using that to predict the green only, if that makes sense. Okay. And that's a really useful, powerful thing for time-lapse imaging because there is so much noise, and noise is random, signal is not. So by capturing multiple time points, you learn more, you get more information. It turned out, I didn't mention this, but our multi-frame output didn't just not flicker anymore. It actually had higher resolution and higher accuracy than the single frame on a frame-by-frame basis. So the model benefited from seeing what was happening before and after, which is logical if you think about it just from like a psychology point of view. The more context you have, the better your understanding is. And that's how these networks work too.
I was wondering what the difference between the Poisson noise and the Gaussian noise was.
The statistical properties of Gaussian versus Poisson noise is what defines the two. Gaussian noise follows a Gaussian distribution. Poisson follows a Poisson distribution. It's as simple as that. And if you look at a microscope textbook, you'll see, or if you look at any microscope tutorial, you'll see that they talk about Gaussian and Poisson noise being components of your microscope image, your digital microscope image. Poisson noise tends to come from detectors, and Gaussian noise tends to come from a bunch of different sources. And the truth is that there may very well be, in my opinion, there may very well be like a certain level of Poisson versus Gaussian noise, that if you added both of those, you might get a good result. But for whatever reason, when we used additive Gaussian noise, we got the best results like right away. And I think that's kind of interesting, that it may very well be somehow a property of the neural network, along with the property of the images, that dictates that additive Gaussian noise is the way to go. Maybe if you're doing a handcrafted solution, Poisson and Gaussian noise is actually a better way to go. Alternatively, maybe it's because we're not just denoising, we're also doing super resolution. Remember, we're going from, we're increasing the amount of pixels in an image by 16x. And if you think about the random gray values of pixels, when you have very few large pixels versus a bunch of small pixels, it could be that Gaussian or Poisson noise, or a combination of the two, will not be able to handle or properly represent the transformation that happens when you have 16 times as many signals. Whereas additive Gaussian does represent that transformation, because now you're making a distribution around pixels, around actual objects of interest. I'm not sure if I'm making any sense, but that's my thinking. But what I think would be really cool is to actually use AI to train a classifier. So then your parameter space is huge, right? You know, you could have a very sophisticated classifier that learns all the things about how images gets degraded. And then you could apply that classifier to all your data.
Yeah, I was kind of wondering what would happen if you combined both forms of noise, the Poisson and the
yeah.
What would happen?
We did try it. I didn't show it, but we did try it, and we didn't get better results.
Oh, okay.
But again, your parameter space is huge. Even with just one parameter, it's huge, because you can go anywhere along an infinite amount of values, right? But when you've got two parameters now you've got to ask, you know, what if you increase one versus decrease the other, and anywhere in between? So I cannot claim that we fully explored the parameter space for Gaussian and Poisson noise. We did explore it some,
Yeah.
but we didn't fully explore it.
Yeah, it'd be kind of cool
And
kind of like a deep fake, the same way those work with like generative adversarial neural networks. If you sort of like train the decrapifier, and then also train the high res until it eventually like I can't really tell the difference. That'd be kind of cool. See what that would
Yeah.
And would improve.
We actually started with a GAN approach because that website that I showed you in the beginning of my talk, where it's called Less Enhance, they used a GAN for that. So our original experiments were all with GANs, and they worked at first on semi-synthetic data, but when we tried to apply it to real-world data, they failed. And what we figured out is that it's really hard to train a GAN, period. But it's really, really hard to train a GAN when you have noise. GANs just didn't seem to deal as well with the noise. Whereas autoencoders like UNets do beautifully with noise. And that's just such a huge fundamental part of our data and our limitations is the noise in the images, that UNets were just so much easier to work with for us. But I like the idea of training a clapifier with a GAN, and or incorporating a GAN into the workflow at least a little bit somehow. I mean, maybe someday we'll do that. One thing I didn't get into is we're training a feature loss model that kind of, to me, reminds me of a GAN, and that we're training a critic to look at the data and be part of the loss function, which in some ways feels like you're just kind of dissecting what a GAN does already, but you're doing it a little bit more piecemeal.
Does anyone else have any questions? Feel free to unmute yourself or text in the chat. If not, I think we can call it there. Is it like 1 more minute?
All right. Well, thank you again for having me. I really appreciate it. If anyone has any follow-up questions or ideas they'd like to discuss one-on-one, please feel free to email me anytime.
Okay, thanks.
Always happy to chat. All right. Thank you very much. Take care, everyone.
Thank you. Bye.
Bye.